All right, check out this picture. If you've ever been to Yellowstone Park, you've seen things like this. This is a hot pot or a hot spring. And look around the edge here. It's all yellow. Just like the yolk of an egg is yellow. For the same reason, sulfur. Sulfur is yellow. And these are sulfur deposits that are the waste product of a bacteria that lives in this hot spring. And this water can be approaching the boiling point as far as temperature. You wouldn't think that anything could live in there. I mean, we actually boil water to kill the things that are living in the water so they don't make us sick. So how in the world could anything live in there? Well, the descendants of the first living things on Earth are able to live in this hot water because they evolved in hot water. When life first arose on the planet, we think that the Earth was still really, really hot about 4 billion years ago. And so these first organisms are able to handle hot water today because they evolved in hot water when they first appeared on the planet. And even if that's not the case, life will find a way. So if there's a way to make a living in any particular environment, life will tend to evolve to be able to fill that niche and live in that environment. So what these are known as are Archaea, which means ancient. And again, they were the first living things, and they're prokaryotes. If you'll recall, prokaryotes are cells, or being prokaryotic refers to cells that have no nucleus or membrane-bound organelles. It's synonymous with bacteria. Bacteria are prokaryotes, and prokaryotes are bacteria. So this caption's explaining the same thing that I just explained. And these organisms that are able to live in very harsh conditions and extreme environments are known as extremophilic or extremophiles. Phile referring, or philic referring to loving. So these are extreme environment-loving organisms. And they're in the domain Archaea. Kingdom Archaebacteria. Now you may be familiar with the taxonomic level of kingdom, taxonomic referring to the classification system that we use to organize life on the planet. You may have heard of kingdoms, but maybe not domains. And so this image is showing the 3 domains. And you need to make this correction on your diagram. The more commonly used name of the eukaryotic domain is eukarya. So replace, this says on yours, I believe, eukaryota. Eukarya is a more commonly used name for that domain. So we now have this 3-domain system that is a taxonomic level above kingdom. So all life on the planet is now classified into these 3 domains. 2 of them are prokaryotic bacteria. And there are these 2 types of bacteria. So it was discovered, the whole reason for the creation of the domain, the taxonomic level of domain, is because it was realized that there are 2 groups of bacteria that are as different from each other as they are from the eukaryotes on this planet. So again, bacteria equals prokaryote. Prokaryote means bacteria. Never forget that. And what that means are cells that have no nucleus or membrane-bound organelles. They're much, much, much smaller than eukaryotic cells. This is a really broad generalization here, 10 times smaller. I mean, the smallest bacteria is probably 100 times smaller than the largest eukaryotic cell, or even more than that. But the idea here is that prokaryotes not only are simpler cells, they're also much, much smaller. There are more prokaryotes on the planet than all other organisms combined. So in other words, if you were to find all the prokaryotes on the planet and weigh them all, in sheer mass, they would outweigh every other living thing on the planet. But you can't see them. So that's pretty incredible to think about. They were once classified in the kingdom Monera. In other words, before the domain system was developed, or the taxonomic level of domain was, it was realized that we needed that. all bacteria, all prokaryotes, were lumped into one kingdom, the kingdom Monera. And at that time, we had a 5-kingdom system. Now we have a 6-kingdom system. So today, the two prokaryotic domains are Archaea and Bacteria, these two. And the two prokaryotic kingdoms are Archaebacteria and Eubacteria. So it can be confusing to keep these names straight, but try to remember that the shorter names are the domain names, and the longer names are the kingdom names. And the kingdom names remind you that we're talking about bacteria here. Archaebacteria, and again, Archaea means ancient. And so they use the term Archaea to refer to this group of prokaryotes because they were the first living things on Earth. 4 billion years, evolved 4 billion years ago, approximately. And then finally, it's important to understand that because prokaryotes were first, they changed the Earth in a way that made it possible for the rest of life to evolve, for the rest of life to even be here. In other words, they set up the global life system, and then we all have evolved and adapted to live within the system that they set up. If you snapped your fingers and did away with the domain Eukarya, the prokaryotes would go on just fine. But if you snapped your fingers and did away with all the prokaryotes, the Eukarya domain would collapse. Everything on the planet would die. One example of that is your microbiome. All the microorganisms, all the bacteria that live in and on your body. If you were to eliminate them completely, you would not be able to survive. So here we see a diagram that should be familiar to you. It's a little nicer diagram than the diagram that we learned about prokaryotic structure and function on previously. But what we're going to use this for is to review. You've already been introduced to the structures and function of the prokaryotic cell in a previous unit. I'm just reminding you here the classification of the organism that we're looking at here. This diagram is representing an E. coli bacteria. So just to practice the classification system, it's domain bacteria, not Archaebacteria, domain bacteria, kingdom Eubacteria, and by the way, Eu means true. So like Archaea means ancient, Eu means true. And they call these the true bacteria because they are the more common bacteria. We didn't even know Archaea existed. We knew Eubacteria existed for a long time, but we didn't even know Archaea existed for a long time. And species Escherichia coli, as you can see why they just like to refer to it as E. coli. What we're going to do now is review the functions of each structure labeled in the diagram, and we're actually going to add some other structures. So you need to, again, you're responsible for both the structure, being able to identify the structure, and describe the function of that structure. So we'll start with DNA, and remember that DNA is found in all cells. In prokaryotic cells, it's not found in the nucleus, right? Because that's what a prokaryote is. By the way, prokaryote means before nucleus, basically. DNA, the function of the nucleoid, the function of the DNA in any cell is to store genetic information. And I just simplified that by saying to store genes. As you know, DNA also replicates. As you know, DNA is transcribed into RNA. So basically the function of the DNA in every living cell, including a prokaryote, are those three things. And all of that is involved in heredity and the functioning of a cell, a living cell. Outside of that, something else that all living cells have is the plasma membrane. It's a selective barrier, if you'll recall. So it controls what goes into the cell and what can leave the cell. And it does that with special proteins that are embedded in a phospholipid bilayer, if you'll recall. That's the structure the general structure of all biological membranes is the phospholipid bilayer. And then there are proteins that give those phospholipid bilayers their various functions, or enhance their functions. All these little round things that we see here in the cell are representing ribosomes. And again, all cells have ribosomes because all cells have to be able to make proteins, right? That's where protein translation takes place. And you know all about protein translation. Outside of that, we have the cell wall. But the prokaryotic cell wall is not the same as as like a plant cell wall. It's made out of a different substance. But it pretty much serves the same purpose, support and protection, and it gives the bacterium its shape, or the prokaryotic cell its shape. And it's made of a substance, or at least in Eubacteria, it has this substance known as peptidoglycan. And you can tell from that name that it's a combination molecule of protein and carbohydrate. So it's a protein-carbohydrate combination molecule that makes up the eubacterial cell wall. And you'll see later when we compare it to Archaebacteria, peptidoglycan's not in their cell walls. Then, of course, we've got something else that's found in all living cells, the cytoplasm. And it's an aqueous solution. And the aqueous solution part of it is what we refer to as the cytosol. But it also contains all the non-membrane bound organelles that you'll find within a prokaryotic cell, which are not many. And that's where the metabolism of the organism takes place. The metabolism of the cell, all the chemical reactions, that's metabolism. The pili are these short extensions here, and they are extensions of the plasma membrane. So they're continuous with the plasma membrane. And they function in attachment. So if a bacterium wants to stay in one place, it will attach to a substrate and stay in that one place. So that's part of their function. But also this process known as conjugation, if you'll recall. A bridge will form between two pili, between two bacteria, and genetic information will be passed from one bacteria to another bacteria. That's conjugation. It's not reproduction. All it is the exchange of genetic information between bacteria. It's also referred to as horizontal transfer of genetic information between prokaryotic cells. So that's conjugation, and that happens across the pili bridge formed between the fusion of two pili of between two different bacterial cells. Flagella are longer, and they spin for locomotion. Remember, locomotion is simply the fancy term for moving from point A to point B. In other words, in the case of E. coli, if it's in an aquatic environment, it's going to be swimming. Eukaryotes have flagella, can have flagella too, but it moves in a different way. It's more like a swimming motion, a side-to-side fish swimming motion, as opposed to a spinning motion that we see in prokaryotic flagella. You need to draw in this structure, and it's just this little ring. And if prokaryotic cell not all prokaryotic cells have plasmids, and if they do have plasmids, there's more than one. I'm just showing one in this diagram. They can have thousands. But all it is a little ring of DNA. It's an extra ring of DNA separate from the nucleoid, separate from the DNA in the nucleoid region. And it's involved in antibiotic resistance in that if the bacteria is resistant to antibiotics, in other words, antibiotics can't kill this bacterium. It's resistant to being killed by the antibiotic, which is what you take when you get sick from a bacterial infection. If a bacterium's resistant, it's because of genes that are on their plasmids. Plasmids tend to have the genes that code for antibiotic resistance to protect the bacteria from antibiotics. The plasmid is also involved in conjugation, along with the pili. So again, conjugation being the passage of genetic information between two bacterial cells across a pili bridge. And the genetic information that is usually passed is a copy of the plasmid. So if you think about it, if you put these two concepts together, antibiotic resistance being on plasmids and plasmids being passed from one bacteria to another, transferred, copied from one bacterium to another during conjugation, that means that one bacteria can give another bacteria the ability to resist being killed by antibiotics. And in that way, antibiotic resistance can spread through a whole colony of bacteria. For example, if you're sick with bacteria, that means there are bacteria growing and reproducing. Their population is increasing in your body. That's what's making you sick. And they can pass the ability to survive being treated, your treatment with antibiotics, and you'll continue to be sick. The antibiotic won't work. The next structure is outward again from the cell wall. Some bacteria, not all, have an outer membrane. In addition to their plasma membrane, they have another phospholipid bilayer that's outside of their cell wall. So again, it's phospholipid, only some. It's used to help us classify bacteria and identify different species of bacteria. The ones that have this outer membrane are gram-negative. They're known as gram-negative because of the special staining technique known as gram staining to differentiate between bacteria that have this outer membrane and bacteria that don't. It's protective and selective, just like the plasma membrane is selective. And then we can have, and again, this is only in some bacteria, this extra layer of polysaccharide known as the capsule outside. And polysaccharide is sugar, so you can think of this as being sticky, and it helps the bacterium stick, just like the pili. Remember, that's a function of the pili too. It helps it attach. But this capsule layer of kind of jelly, sticky jelly, can allow a bacterium to stick, for example, in your lungs. If you have pneumonia, it's an infection of your lungs by bacteria, and the bacteria that cause pneumonia have a capsule, and this helps them stick in the lung tissue. And you're trying to cough them out. Well, you can't cough them out because they're stuck in your lungs by the capsule. So again, only some have the capsule, but it tends to be the kinds of bacteria that make us sick. In other words, pathogens tend to have a capsule because it helps them infect us. So it's both protective and sticky. So this E. coli is acting as our representative of Eubacteria, and in general, Eubacteria are more diverse and occupy more habitats than Archaebacteria. Again, U means true because they were the most common kind of bacteria until we discovered Archaebacteria. But this is a generalization, and it may change as more prokaryotic species are discovered. In other words, as we discover more Archaebacteria, which is happening all the time. Both new species of Archaebacteria and Eubacteria are being discovered all the time. So these generalizations, that kind of generalization may not hold up forever. Okay, on this diagram, well, now let's take a look at, if that's Eubacteria, then what are Archaebacteria? What makes them different? And this slide is about that. And so here we're looking at a phylogenetic tree, a representation of the three domains again, just like the first slide of this slide series. And we start down here at the bottom of the phylogenetic tree with what's in the key here is the presumed common progenitor of all extant organisms. There are some, heavy scientific words there, but progenitor meaning that's where life got its start, and extant meaning of all living species. Extant's the opposite of extinct. So we often refer to this as LUCA, the last universal common ancestor, LUCA. And as we go up in the tree, we see that bacteria branch off first, and remember that that's kingdom Eubacteria, right? So it's domain bacteria, kingdom Eubacteria. And then Archaea, or kingdom Archaebacteria, branch off later on, and then Eukarya, which includes kingdoms Protista, fungi, plantae, and animalia, branched off and have common ancestry with Archaea and Archaebacteria. And there are some reasons for that. We have some similarities, some commonality that suggests that Archaea and Eukarya are more closely related to each other. So let's take a look here. The problem is that Archaea, and when I say the problem, I mean like when you look at Archaea under the microscope, they look a lot like Eubacteria, so it's difficult to tell them apart. The difference is in their ultrastructure. In other words, their molecular structure, really, you know, what the molecules that are making up the cell, and in their metabolism, the chemical reactions that are taking place to keep them alive. Archaea cell walls have no peptidoglycan, so that's one major difference that we can use to identify and differentiate Archaea from bacteria. What they do have are these things called S-layer proteins. I just threw that in just in case you're wondering, well, if they don't have peptidoglycan, then what do they have? Well, they have these special proteins that are known as S-layer proteins. The plasma membranes have different lipids. And the reason for that is because if Archaea tend to hang out in harsh environments, and these different lipids are going to help them survive in these different harsh environments. Archaea genes have exons and introns. Bacteria genes do not have exons and introns. And if you'll recall, exons are portions of genes that are going to remain in the messenger RNA and be transcribed into translated into protein, whereas introns are cut out of the messenger RNA. They're not included in the messenger RNA. They're not going to be translated into proteins. Archaea have these, and bacteria don't. So that's one of the things that suggests that we're more closely related to Archaea, because we have exons and introns in our genes. And again, like I just mentioned, they have these differences to survive harsh environments. They're extremophiles, this term here, extremophiles. So examples of extremophiles are methanogens, and they live in anaerobic environments. In other words, what's extreme about the environment is there is no oxygen. Anaerobic means no oxygen. Like mud. If you just go down a couple inches in mud, there's no longer oxygen in that mud. The oxygen gets all used up by the bacteria that are living down there. So only the bacteria that can survive without oxygen are going to be able to live. Anaerobic bacteria. And they're called methanogens because they produce methane. So if you've ever gone like walking through a swamp, and with every step it releases a smelly gas that kind of smells like rotten eggs, and other things. The rotten egg smells hydrogen sulfide, but methane's also in there too. So here's an example. Methanobacterium over here in Archaea. Methanococcus. Halophiles are salt-loving. Halo refers to salt. And they live in hypersaline environments. So again, over here we have Halococcus and Halobacterium. And then thermophiles are heat-loving, and some live in boiling water, like that image we saw on the first slide in this series. So Thermococcus here, Sulfolobus, excuse me, and notice the sulfo, so that's probably the organism that's living in that hot spring on that image that we started out with. Now that's not to say that there are no bacteria that are also extremophilic or extremophiles, because notice here, Thermatoga is an extremophilic bacteria, or Eubacteria. So, like I said, life will find a way, and notice that the bacteria, Eubacteria, are very, they evolved early on, as did Archaebacteria, in a very harsh, hot environment when the Earth was first cooling down. So these may be descendants from species that evolved very early on, and that's why they're branching off very early on in the branch for bacteria here. And then there's one other group that's out here in bacteria that isn't represented here, but it's such an important group that we can never ignore it, and that would be cyanobacteria. Cyanobacteria, if you'll recall, are photosynthetic bacteria, and they were the ones that initially put oxygen into our atmosphere, started building up oxygen in our atmosphere, and they continue to supply most of the oxygen in our atmosphere today, or at least a very large part of it.